real pleasure to introduce Rick Young today for our seminar series. Rick's a professor of biology at MIT, member of the Whitehead Institute, and is a real thought leader in the fields of systems biology, of gene regulation, and a longtime innovator of genomics technologies. He's got remarkable energy, always finds ways to the most exciting and timely science. Kind of feels like he's had about 8 scientific careers now. I first met him around 2000. Rick was, at the time, a leader in the field of yeast genomics. A couple years later, he published seminal work on the master transcription factors of human embryonic stem cells. Then he taught us about transcriptional amplification by the MYC oncogene. He taught us about super enhancers, topology, and now he's thinking about condensates and IDRs. Along the way, he's sort of trained leaders in the field and spun out very many companies. As you might imagine, he's received many accolades and awards over the years, and I'll just highlight that he is a member of the National Academy of Medicine and the National Academy of Sciences. Amazingly, despite all of these activity and accomplishments, Rick has enough energy for life outside the lab. As many of you know, he's a commercial pilot, and he regularly risks his life on sort of perilous backcountry treks across wild frontiers like Iceland. I was excited to tell him that I was going to go to Iceland, having been inspired by his stories of Iceland, but he told me that Iceland was yesterday, and now you need to go to Greenland, and that he's taking a dog sled somewhere, and we'll be staying in tents at minus 20. Iceland's enough for me. Thanks, Rick, but enjoy. Get back safe. Rick's also an amazing speaker. And I think it'll be really great that we have you here today. So thank you for coming. I'll turn it over to you.
Thank you, Brad. It's really a pleasure to be across. Yeah, it's a lot of pressure to be across town here in this home. I have been in this environment now for 40 years, and it's so nice to come over to Harvard and talk to you about these subjects. My title bears some resemblance to what I am going to talk about. What I really want to talk about is a combination of things. I want to talk about, in cells, how proteins move from the stoichiometric complexes that we've all learned and are in the textbooks to larger assemblies that are functional in cells, provide cells the ability to function in the ways that I think we now understand. And I'm going to tell you that those assemblies create distinct chemical environments, and cells exploit that to regulate function. I'm going to transition to what I think of as these types of assemblies that occur at key genes that are regulating cell state. They're dysregulated in cancer. We're getting closer to what I would call the molecular architecture of these assemblies. And then I want to come back to what I think is the therapeutic opportunities that emerge from these concepts. So to do this, I'll first talk about these functional assemblies. I'll then talk about oncogene regulatory architecture, and then the therapeutic implications. And these are my conflicts. So the way I learned to think about cellular compartmentalization is still true. There are membrane-bound organelles throughout the cell. There are signal sequences in some proteins that help them get there. And that view has enabled much of my thinking about how cells work. But we now know from investigations made by experts in each of these functional areas that at least part of the lifetime of every signaling pathway, every transcriptional event, every DNA damage repair event, and our cognition involves the formation of non-membrane assemblies. So some of these have very long lifetimes. The nucleolus is a pretty stable non-membrane entity within the nucleus where pre-ribosome biosynthesis is being done. But most of the molecular events that have fascinated me, transcription, splicing, signaling, are occurring in very short time frames. These assemblies can occur in seconds and then dissolve. And the issue that is raised by this history of molecular biology comes from the really inspirational training we've gotten from structural biologists who've helped us understand at atomic resolution the structures of proteins. And here are some of my favorites. This is the Micmacs heterodimer on DNA solved by Steve Burley many, many years ago, and the histone octamer. And this view really does dominate our thinking as we consider what regulatory biology and what's gone awry in regulatory biology. But the business end of most of these molecules that I'm interested in are not structurally stable. They're made up of low complexity domains. They are classically called disordered sequences, and you see them, for example, in the effector domains of all transcription factors. All transcription factors have effector domains that are intrinsically disordered, and there's no crystal structure that has ever been determined for those domains. And similarly, the model that we all examine in all its glory for the histone octamer is missing these N-terminal tails on which all the modifications are done that confer some kind of stable or quasi-stable epigenetic state on those portions of chromatin. The one little biophysics lesson I want to give you, and apologies to all of you who know much more than what I'm displaying here, is that we need to move a little bit from the classic view of shape complementarity as the driver of interactions in cells. Because if it were just shape complementarity, lock and key, we would not be the dynamic non-equilibrium organisms we are. We would be very clunky, static organisms. And so the 25 to 30% of our proteome that is these disordered regions provide proteins in cells with mechanisms necessary, chemical mechanisms, to use large numbers of low affinity interactions to assemble. And these assemblies can occur, interestingly, with very high Hill coefficients. They can occur almost digitally as you cross what we think of as a phase separation boundary. Now, polymer chemists have known this since before I was born. This is an old field, but in modern molecular biology, we passed it over for many years in order to build a more foundational model of how things work with shape complementarity. That's not, however, how I think transcription works. I think the way the transcription works is not a set of individual stoichiometric interactions built like a Rube Goldberg apparatus where each has one contact with something else. But rather, there's these events that coalesce proteins and RNA molecules at DNA sites. And what you're seeing here on the right is pretty much a real-time movie of the transcription apparatus forming these assemblies. And these assemblies are occurring on enhancers as they interact with promoters. And there's a nice correlation between the size of the assembly, its longevity, and the transcriptional output that you see. So the smaller assemblies are very unstable and have 1 to 2 second time frames. As you get larger, those assemblies can have minute or many minute long time frames. And that's where large amounts of transcriptional bursting occur. So these kinds of assemblies can do chemical work for you. And as the transcription apparatus moves from its site of initiation in such an assembly, a couple of things happen. One thing that happens is in that burst of RNA, those negative noodles create a high density of negative charge and will dissolve those transcriptional condensates. So it's a nice feedback mechanism that we believe occurs and gives you this ability to form the assembly, and then when you transcribe, you dissolve it. So this kind of feedback regulation is an elegant way of taking the product of the reaction and causing it to shut you down. We also believe that the cyclin-dependent kinases that are phosphorylating the C-terminal tail, this intrinsically disordered region of 52 repeats of PTS-PSYS in our RNA polymerase II molecule, that's just changing the chemistry. And what we've seen then is the phosphorylated C-terminal domain will now move away from, it has a different chemistry than that initiation assembly. And it acts as a chemical antenna to attract the splicing factors that must be there very quickly, because much of splicing occurs co-transcriptionally and can occur within seconds of a polymerase moving through a splice junction. So this hyperphosphorylated C-terminal tail now has a new chemistry that attracts these RS-rich proteins. So you can do regulation. But what's really responsible for the 5 to 10 billion proteins that you're synthesizing in each cell that are then moving across the cell to sites where they're going to functionally occupy these compartments and do work? How do they get there in an efficient fashion? Well, classically, based on early work from Blobel, that would be a signal sequence, a small set of amino acids, maybe only 6 to 10 amino acids long, that would be a docking site that would facilitate the transport of a protein through a membrane, say, across the nuclear envelope. It could also be that since most nuclear proteins don't actually have these sequences, that they could assemble preferentially in the nucleus at the right site by having a high affinity docking partner. And I'm sure that's true. For example, you can have heterodimeric transcription factors where only one of the two proteins has such a sequence. But this can't possibly explain the efficiency with which those 10 billion different protein molecules all sort themselves out into functional sites and do work with the efficiency that they do. And so we came to imagine that what's doing that is a distinctive compartment chemistry that is engendered by the amino acid sequences in proteins. And the first line of evidence that we had that this was the case was we took the 15% of FDA-approved drugs that are naturally fluorescent and distributed them on cells. And this is an example of an experiment with live HCT116 cells. And what you're looking at are images where we've just highlighted in each image a nucleus. And you're looking at either in the top left, a bright field image, or throughout the rest where you're seeing fluorescence of that FDA compound. And what you see is distribution in all kinds of different places. You see enrichment in membranes, you see enrichment in compartments, you see enrichment in subnuclear compartments. So for example, camptothecin, a good inhibitor of topo2, you'll see in the upper center there that it's concentrating in the nucleus, to some degree in nucleoli. This is good because topo2 is in the nucleus, and so it's concentrating in a body where the target lies. But you also see many other drugs like sunitinib in the bottom right. That's an RTK inhibitor. That concentrates in nucleoli. That's also a very toxic drug. I don't believe it's really an RTK inhibitor in vivo. And so forth, you see all of these drugs concentrate in different portions of the cell. And if you're a drug developer, you'll recognize that when you look at distribution, it's a tissue distribution you look at. You don't do experiments like this to ask, oh, and once in cells, where does the drug go? So a chemist, Henry Kilgore, in my lab went a step further, and he imagined that if these compartments have a distinctive chemistry, then because like solvates like in chemistry, each individual protein molecule had to resemble in some way the chemistry of the assembly into which it fell. And so he took what he called scaffold proteins, the dominant proteins in each of these condensates within cells, and he made little droplets in vitro. And then he took a large library of fluorescent compounds and asked, what is their partition coefficient in each of these single protein droplets? And then he used a machine learning algorithm to ask, are there shared functional groups on these small molecules that might explain why there's preferential assembly of preferential concentration of some drugs over others, and found from that algorithm that he could make those predictions. But remarkably, what he found that I just didn't believe was that the predictions in vitro of small molecules held in vivo. The majority of the small molecules that would concentrate in, say, a splicing factor droplet in vitro would also concentrate in a splicing factor condensate in the living cell. And I didn't think that would happen because in the living cell, that's such a more complex compartment. But as he pointed out, the chemistry has to have a shared feature. And so this led us to believe these compartments have separate chemical environments, and the separate chemical environments at least lead small molecules to selectively partition in specific compartments. So that led to a wonderful collaboration with several folks in Regina Barzolet's lab at MIT. So Regina runs one of the most sophisticated deep learning labs at MIT. We were fortunate to connect with her and to ask, is it possible that proteins harbor a chemical code that also causes them to selectively assemble in specific compartments? And so what we did is we took a large language model called ESM2. There are a variety of these models. AlphaFold emerged from such a model. And we asked if we took proteins that had been annotated as being part of various compartments, and this emerged from a variety of data sets, for 12 different condensates, 12 different non-membrane bodies in cells. And you train these models using what's called a transformer if you don't want to use exceptionally large amounts of data like you might have for the English language on the internet. You can use a transformer, a feature that was invented in 2019 that allows you to use less data than you'd imagine might be needed for training. And so 70% of those protein sequences were used for training, and then 30% were withheld. And so the key question is, when you do that training, will the model, and we call this model PROT-GPS, will the model accurately predict where a withheld protein goes? And I was astonished to see that, in fact, the prediction performance is extremely high. These numbers for AUC ROC are exceptional for a large language model. Many of the predictions for these withheld proteins were on the order of 0.975. So that says that the algorithm can detect something about the amino acid sequence that is a pattern that prefers the chemistry in one compartment versus another. But what we really wanted to do is to see then, and actually this is what the reviewers really wanted us to do, so could this just be explained by human observation? Could you just go into proteins that were predicted, say, for a nuclear job and see that they had nuclear localization sequences? And most nuclear proteins don't contain them, so that's not a good explanation for the chemistry. But for those of you who study splicing, SR is a common dipeptide in splicing factors, but it turns out that that's actually more frequently observed in the proteome than in nuclear speckled proteins, so that didn't help us. We did see these famous FG repeats in nuclear pore proteins, and so that the algorithm did recognize that. And the key question for this chemist in my lab, Henry Kilgore, was, but everything has a physicochemical property, so you can describe each amino acid as having a particular physicochemical property. Was that all that was responsible? And he and others built a variety of models to try and detect that, and could not find that the algorithms that were built on physicochemical features that are common in textbooks would explain this behavior. So the bottom line is the algorithm's detecting something that is not as simple as the terms we use to describe the physical chemistry properties of amino acids. Now, an important test of whether or not an algorithm is actually seeing something is whether or not it can predict something novel and have that novel thing, in this case, go to a compartment. And this is not a very, in the world of computing, these so-called generative algorithms are notoriously unsuccessful. But this ProGPS algorithm was exceptionally good at predicting proteins that were novel, that could be appended to mCherry, that would take those proteins into the nucleolus. It was okay at predicting proteins that would go into these splicing speckles, and it was terrible at many of the other tasks that we gave it. We have a detection issue as these condensates get smaller and smaller, and as they get more and more transient, then the signal-to-noise issues for us are terrible. And so we don't yet know how much of our failure for some of these smaller compartments is a technological issue or an issue with the ability of this algorithm to detect the chemistry. But there was another question we could ask of it. Could we go to ClinVar, get all the pathological mutations, and ask what subset of them are predicted by ProGPS to cause the protein to go to the wrong compartment? And so we had a collection of those. I'm showing you a small subset of the experimental data for 20 tests that we did. And in 17 of these tests, in fact, the variant proteins altered their cellular distribution in a manner that was predicted by the algorithm. In 3 cases, that was not true. So we think that each of these subcellular compartments, which are assemblies of multiple polymers, has distinctive chemical environments, and those chemical environments are facilitating how a protein moves across the cell to assemble with its drinking buddies. And by the same virtue, proteins that don't like the chemistry of a body will cause it to spend far less time in the vicinity of that body and effectively give a protein a shorter path length to the assembly where it's going to function with its friends. These are contributing to normal regulatory biology. I showed you one instance where the phosphorylation of RNA polymerase causes it to prefer to be associated with a splicing apparatus. And I just showed you cases where we think pathological disease mechanisms are engendered by a change in the location. So does this offer new therapeutic opportunities, which was my teaser in my title. And first I want to tell you about work, just very briefly, that we published a couple of years ago, where we discovered that cisplatin is not working by randomly platinating DNA in tumor cells, as is the textbook model, but rather it concentrates in large, stable transcriptional condensates and platinates the oncogenes that have acquired those superenhancers. And so we think that despite them being a chemotherapy and quite poisonous, the reason they have efficacy in a broad distribution of cancers, and the reason that family of drugs is used in 1 in 5 cancer patients across the world, is because they're destroying the driver oncogenes of each cancer due to this feature that the oncogenes evolve large superenhancer assemblies. And those assemblies sit there for minutes or hours concentrating cisplatin. And so now the on-target drug concentration for that oncogene is extremely high compared to anywhere else in the genome. So that makes me more and more interested in understanding these odd oncogene condensates. The superenhancers associated with most of the master transcription factors in all our normal cells tend to be about 10 or 20 kb in length. They use that much DNA in these assemblies. The superenhancers that drive MYC and other oncogenes are 100 kb or 200 kb or 300 kb of DNA. They're an order of magnitude, they occupy an order of magnitude more DNA. So what can we learn about the regulatory architecture of these structures? Well, what do we already know is a good question to start with. And we know most genes are regulated by multiple enhancers. Most all of our genes have multiple enhancers. Some of these engage in very long-range contacts with promoters. Many of them are within 10 or 20 kb of a promoter, but enough of them are a megabase away to make these very big distal and important contacts. They're bound by transcription factors, probably defined by the transcription factor components. They're recruiting coactivators, defined as transcriptional components that don't bind DNA. And even enhancers are bound by RNA polymerase II and are transcribed. So these assemblies form active transcriptional compartments, we believe, and most cancer risk variants, GWAS, are occurring in those enhancers that we're talking about. So that's known. And the other things that I wish to learn are along the lines of, well, what are the precise transcription factors that are binding to these enhancers? What are the roles for RNA? I told you these enhancers are transcribed. The RNA at enhancers is probably a better determinant, probably a better surrogate mark of an active enhancer than anything else. And what are the mechanisms of these cancer risk variants? So we got together with Anders Henson and Virat Goel at MIT, and we adopted a technology that Anders developed called region capture micro-C. And in effect, what this technology does is it takes micrococcal nuclease high C and oligos that capture a region, in this case about 2.8 megabases, and it allows you then to focus all your reads in that region and get very high density information. What Anders has already described is 3D structures that he thinks are being formed through what he calls coalescence, which I will just call a condensate. And because the read depth is so great, you can actually see, Martin Airy convinced us, you can actually see the footprints of individual transcription factors that have been protected from that digestion. So let me show you an example of this. This is the MYC Oncogene and HCT116 cells. These little semicircles represent the contacts that come from 2 billion sequencing reads. So these are about, we can drive them into about 400 loops that just represent the contacts that DNA-bound factors are making. And so I'm going to focus your attention, at least in these cells, on this 0.5 megabase microcompartment that's highlighted in green just upstream of the MYC gene. This is where we already knew there were many clustered enhancers in these cells. And so I'm annotating here that region in green. We'll just call it an upstream microcompartment. What you can see are the region capture micro-C loop regions annotated in green, and you see them associated with encode enhancers. So the orange lines are encode enhancers, and the algorithm that we developed more than a decade ago that just takes collections of clustered enhancers and for simplicity calls them a super enhancer is also annotated here. And what you can see at the very bottom is that the long-range interactions, the long-range 3D interactions, are really dominated by those clusters of enhancers and the promoter region around MYC, all of what you'd expect. But you could also imagine that this is a model for the collection of many different cells, as a composite model for the collection of many different cells, where that MYC locus has folded up with transcription apparatus and DNA. But where I wanted to go to is what transcription factors are defining these enhancers. And so what we were able to do is a kind of a classic thing you do with micrococcal nuclease protection. You identify sequencing reads that were protected. You can do what's called segmentation. You basically decide what the boundary is. And then you can go to existing data like ChIP-seq data, motif data, and make predictions about the transcription factors that are bound at that site and actually to the specific motifs that have been determined in the past. And if you do that, what you see, I'm just giving you an illustration of a tiny, tiny fraction of this active locus for an enhancer that ENCODE described as E36 in this region. So it's one of about 60 enhancers. It's in the center of a region that we'll call a region capture micro-C loop region, where there are many contacts with the MYC promoter. And we can see then these footprints that range in length from about 6 to 250 base pairs. The median size is about 40 base pairs. The average DNA length separating these TF footprints is about 150 base pairs. It is sometimes the case that a nucleosome footprint is seen at that site and is probably responsible for that separation. So this is what a footprint density map looks like, where the read length is plotted on the y-axis, and across the x-axis, we're looking at these segmented footprints. So when we take ChIP-seq data, so evidence of binding, and we annotate that onto these footprints and look for the motifs, we generally find the motifs. And this is just a map of what these motifs look like. And one of these days soon, I hope to actually show you a kind of a base pair resolution model of how each of these transcription factors is bound to these motifs. But this is what we have at the moment. So we think we can identify the precise transcription factor bound structure of enhancers, at least version 1.0. And what I want to tell you briefly is whether or not we can identify roles for RNA in this compartment architecture and function. And so we've found and reported about 2 years ago that most transcription factors have an RNA binding domain. So classically, transcription factors have a DNA binding domain. I told you at the beginning, it's highly structured, very stable, and an effector domain. But what we realized was that we had missed a non-classical RNA binding domain because we were depending on the RNA binding domains that were structured and defined by splicing factors and many other RNA binding proteins. This RNA binding domain in transcription factors is typically adjacent to the DNA binding domain and is essentially identical to HIV TAT's RNA binding domain. So HIV TAT is TAT's transcriptional activator. It binds to an RNA sequence called TAR, and it uses this 9 amino acid arginine-rich motif to bind that TAR sequence. This sequence, in the vast majority of human transcription factors, is the same sequence, and we found has the same activity. It binds RNA with the same affinity that RRMs, these classic RNA binding domains, bind RNA. And if you delete it, the transcription factors no longer like to dwell in spaces that look like they're these transcriptional condensates. So we were very interested then in what would happen if we got rid of RNA in these cells. So we treated nuclei with RNAs. And as we expected, all these transcription factors that have RNA binding sequences are depleted. And moreover, when that happens, what we lose, and this is the very bottom line in this image, we have an RNA sensitivity score. What we lose are all those long-range contacts. So RNase digestion causes a change in both transcription factor binding to these sequences and in the contact frequencies that occur. So we think RNA is important for the structure and maintenance of these assemblies. I want to just say a few words about interpreting disease-associated genetic variation because UC Taipei did this beautiful study that drew us in these colorectal cancer cells to a variant, actually a collection of variants that elevate risk for colorectal cancer. And so what you're seeing in the second line there are these colon cancer GWAS SNPs. I'm going to focus your attention now on the zoomed out region, this 1 kb region, centered on the index SNP, which Taipei showed was a binding site for TCF7 like 2. Now, the TCF factors are the binding factors for HDACs in the absence of Wnt signaling. But with Wnt signaling, you get beta-catenin accumulating, goes into the nucleus, and it displaces the HDACs and gives you elevated transcriptional activity. And what we were able to find in this footprinting is actually the exact site where TCF7 like 2 binds and engenders this specific risk for colorectal cancer. And we could see how that site bound by that transcription factor contacts enhancers upstream in that cluster. So this site is just outside of an ENCODE annotated enhancer, but is physically connected to the enhancers in that locus. So that helps us understand how that collection of SNPs creates a marker for an index SNP that looks like it's the functional SNP, which is functioning by elevating the binding affinity of TCF7 like 2 and bringing more Wnt beta-catenin signaling to the MYC locus, which is known to be the function of beta-catenin in elevating MYC expression. So what have I told you so far? I've told you that genome regulatory architecture is really defined by these transcription factor assemblies. The transcription factors are binding specific sites. We can define what they are now at base pair resolution. We can also look at ChIP-seq data to see what the other factors are that are bound there. We know now that RNA is not just a bystander component useful for charge balance in these condensates, but in fact, it's an important regulator of the stability of the transcription factor assembly. And we think this kind of data can facilitate our interpretation of genetic variation in these regulatory DNA molecules. So I want to go back to a subject that I kind of started at the beginning and just go through some therapeutic implications and talk to you about data that is not entirely public. So I started out by saying that I think most cellular functions are occurring through the formation of these non-stoichiometric assemblies in cells, that those produce a chemical environment that's distinctive for each function. And I just gave you an example of transcriptional assemblies, which we look at as having one flavor of this chemistry. And so what's evolving, I think, is an opportunity to target the condensates themselves, in particular to target those condensate assemblies that represent dysregulation in cancer. So there are a number of ways to do this. We can exploit dynamics, and I suggested to you that that is actually how the cisplatin family works. That is to say, cisplatin is going to concentrate in any transcriptional condensate, in normal cells or in tumor cells. But the difference is that that condensate in tumor cells sits on oncogenes stably in a form that is going to recruit most of that drug. And then cisplatin is going to intercalate and platinate that sequence. So we're already exploiting the dynamics of these compartments for one of the most widely used chemotherapies. How about if we could eliminate condensate dysregulating proteins? Well, that is what's happening in APL, one of the few cancers you can cure. And you can cure it with a mix of arsenic trioxide and atra. And that's being done because, in part, PML, as part of that fusion, that PML-RAR alpha fusion, is coordinating the arsenic trioxide and helping drive it to become sumollated and then ultimately destroyed, allowing that PML body to reform, and now those cells are differentiated and they'll die. And so that cure, I think, is a cure that comes about because you're eliminating a dysregulated oncogenic fusion protein that is altering that PML condensate. I've seen data now from DuPont. It's public. They've presented posters on this. They have drugs, small molecule drugs, look like they're going to be in the clinic by the end of the year for both beta-catenin and for MYC, where tumor cells that are overexpressing those proteins, the drug causes them to form an inert depot in the nucleus away from genes they normally regulate in their oncogenic state. These drugs are highly tolerated. They're oral drugs, highly tolerated, because you need the excess concentration of the protein in order to get that phase separation to occur in the presence of the drug. So the drug doesn't do anything to normal cells that have normal levels of beta-catenin or MYC. And so you're exploiting a biophysical property of these condensates that are forming in the context of excess expression of these oncogenic factors. And I think this will be a general approach in many cases where we're interested in taking advantage of excess production of transcription factors, whether they're normal or fusion factors. And then we're working on a second generation of ProGPS. That's this large language model algorithm we're using to detect and predict where proteins go in models. Because as I told you now, we can see the chemistries that prefer to go into specific compartments. It's the small molecule chemistries. So this should give you the opportunity to take a small molecule that has a warhead, a small molecule that can bind with high affinity and selectivity to a particular protein target, but now concentrate it in a body where that target lives. And in doing so, vastly increase your therapeutic index. Keep that small molecule away from off-target interactions that all small molecules have. So I think that algorithm, should we be successful in predicting how the chemistries that are formed by these RNA, DNA, protein polymers, and how they like to concentrate specific functional groups in chemistries, I think that gives us lots of options for lead optimization. This is the crew that did this work. Thank you for your attention.